and welcome everyone to this week 3 live hands-on lab screencast review section. I hope everyone is happy, safe and well, and that you can join us in good spirits today. So in this live screencast we're going to go through, of course, let me pull up the right slide here. We're going to obviously go to our class website and find the week 3 content that we're going to focus on today. Here it's all about sequence alignment, the fundamentals of how biomolecular sequence alignment work, the algorithms and the applications of sequence alignment, which is such a fundamental topic, of course, in bioinformatics and underpins much of what we do in bioinformatics, whether that's genomics, as we talked about in our lecture videos, or structural biology and structural bioinformatics and drug design and drug discovery. We're often doing alignments to compare sequences or structures or other things using these methods that we talked about in those videos. So find our week 3 content, then of course scroll down, find the lab hands-on sheet. Again, for this week, this is a PDF that you'll be able to fill in and put your name, your UCSD number on it, etc. Again, please do remember to download. This should say week 3 here, not week 2. Please do remember to download this PDF file and open it in your, if you're on a Mac, open it in a software like Preview that comes pre-installed, or Adobe Acrobat Reader if you're on a PC or similar. And then we'll all be on the same page. We'll have that file to upload. So now there were only 3 required sections for this week's lab, and then there were some extension sections as well for those students that were particularly motivated or will end up working in this area in the future, that delve a little bit deeper to the forefront of the field with some of these more advanced methods using profiles and hidden Markov models, and also using structure to inform our investigation of these distant evolutionary relationships. So we're really going to focus here today on these first, we'll focus on the first 4 out of these sections. These are all that you absolutely have to do to fill in to get the full marks for this lab section. And we'll take it pretty easy. We're about 15 minutes for that first section where we go visit dot plots. Remember, dot plots were this simplest of visual approaches to alignment, and then we'll dig into those dynamic programming approaches. I'm going to pause here before we get started for any comments or questions. I'm going to make sure that this stream is working because I don't see any comments coming up yet, and so just bear with me for 2 minutes while I see if folks are here and available. So again, on the logistics side, similar to last week, you can add comments here on YouTube, on the YouTube Live section, but we're also over on Piazza. We'll be able to see those comments as well. Okay, thank you. Thanks for the heads up. So I'm going to go over now to the bigger screen here and look at the PDF. So in the PDF here, let me focus in on it. Here where it talks about sections 1, 2 and 3 has been absolutely required. They're first going to delve into these dot plot approaches, and then the Needleman and Wunsch alignment approaches, and then 4 to 7 are delving into SciBlast, hidden Markov models, and the gold standard for this approach, which is using structure if you have it. So in section 1, it points you to these 2 URLs, and these URLs will take you to the same thing. They're just little mirrors of each other in case too many people are playing on one at a particular time and it ends up being a little slow for you. Feel free to use the second link. That'll take you to another iteration of this thing. I'm going to click on these 2 here, and that'll open up this little dot plot app. And remember, this dot plot approach now should be familiar and pretty straightforward. So let me go here and make sure we're in the right place. Okay, so here we have, it's a bit, let me zoom in a little bit perhaps. Okay, so I have two figures here, two plots of the same size, dot plots. One is for protein on the first panel here, and the other one is for exactly the same length. It's actually that corresponds to the same sequence, only it's the nucleotide version of this sequence. So they're all about, just under 200 characters long, whether that's amino acids or nucleotides in the second one. And there's a dot placed everywhere where a character matches, where there's a character correspondence. Okay, so let me actually make myself a little bigger here so we can see how that goes. Okay, that's better. Now I've got my head in the dot plot, right? Okay, we can move it out of the way. So the idea here is that there's some sliders on these windows. Let me see if you can see where my mouse is. So my mouse is here on this first slider, and if I increase this slider, the dot plots change appearance. Hopefully you're able to click on these links and see this. We can both change the window size and how the window moves, right? So here, hopefully you can see these plots look different. Okay, how are they different? That's what I want you to think about. And then there's also this match stringency. So this a combination of these parameters here. For example, if I increase the match stringency to be very strict, so here what I'm asking with this set of parameters, I'm saying we're going to consider. Let me close the window for a second. That's the physical window, not the match sliding window. But what we mean by the window size here is rather than asking for one character at a time to match, to put a dot in, we're going to look in a broader little window of characters, in this case 3 characters at a time. So if that's amino acids, it's going to be 3 amino acids. And we're saying all 3 of those amino acids must match between the 2 sequences, the one on the vertical and the one on the horizontal. And obviously that means there's less dots. So it's filtering out, it's an approach to try and filter out spurious matches and focus in on the signal here. So a noise reduction kind of protocol. So the questions that I have are, well, first off, regardless of what parameters we use here, so here I've got pretty clean looking 2 plots, but there's still more dots in that second plot, in the nucleotide plot, than there are in the protein plot. Why is that? Right, so what I wanted you to think about is that, and in discussions, we often hear students think, we know a lot of biology, we know a lot of background, we think, well, the codons that we dealt with in the last lab, takes 3 of those nucleotides to encode a single amino acid and all sorts of things, and that's correct, right? But it's not what we're looking for here. That's too much. This is a much simpler, more simpler-minded question here. What we're really showing is the intrinsic alphabet size of these 2 character types. So what do you know about protein? What's it made up of? Well, it's made up of amino acids, right? And how many natural amino acids are there? Well, there are 20 naturally occurring amino acids. So that means we have an alphabet of size 20 in this case. What do we know about nucleotides, or the DNA sequence that we have here? It's made up of 4 nucleotides. So there's an alphabet of size 4. So it means when you're drawing from a pool of just 4 things versus a pool of 20 things, you're much more likely in this case to have matches just by chance, because you're only drawn from 4 possible characters versus the 20. So that may seem, just a little side note and an interesting observation, which it is, of course, but it's also tells us something important, something that we should pay attention to. It means that there's actually more signal in the biomolecule with the larger alphabet. There's more signal in protein. So that means if you can translate your query when you're looking for genes or nucleotide sequences or any of those things to amino acids, to protein, you should probably do it, because you're going to get much more signal and less noise, less chance for spurious matches in this case. So, that second question here, we can go to our PDF. You'll see that there's a place to enter in these sorts of these answers. So let me pop over to the PDF for a second. So if we're here, you see, why does the DNA sequence have more dots than the protein sequence? The reason here is the intrinsic alphabet size, intrinsic composition. Put this in your own words and think about it a little bit more deeply. We can talk about these things on Piazza and try and explain this in more clear ways if it's not making sense to you. But we're talking about the intrinsic composition of the alphabet, of the 4 nucleotide bases in DNA versus the 20 amino acids in the protein sequence. So you should be able, just to write in this PDF document, add as much details as will be useful for you in the future, because these will be notes for you as well, of course. The second question here it asks, same as it lists on that on the website, how can you increase the signal to noise ratio? Well, you increase the window size and play with the match stringency, make it more strict. Question 3. What does a match stringency larger than the window size yield? Well, I haven't played with that yet. Let's go back and see if we can do that here. So I'm back over here and I'll make sure I'm on this. So it says, what can the match stringency larger than window size yield? So here the match stringency, I'm going to increase it. Oh, what happened? Well, it gives me this error message, this red text up here, and normally, of course, if I was coding this, I would catch this and give a useful message to a user and say, please don't do this. But I did this on purpose. I left it with this vague kind of error message, right? Which isn't helpful at all, actually. It doesn't tell us what's going on. But what do you think is happening here? Well, we're setting a window size, in my case, 3. That's the first slider. And then I'm asking for, out of those 3, we need 6 characters to match. So that's silly, right? That's a stupid thing to say, because we don't have 6 characters in the window to match. So of course you can't never match 6 characters. You're going to have a blank plot here, or in this case, an error message returned. You're going to have either 3 for this thing to work, or less than 3 match stringency. So match stringency is our threshold of how many characters within the window you need to match. And we'll come back to this, or we did in our lectures, our video lectures. When we used BLAST and the heuristic, where we're using these words and these lookups, you can miss things because your word size and your stringency is too severe for the problem. If you're looking for small motifs or small sequence fragments, you're probably going to have to change the window size in the BLAST parameters, or you'll miss things. So we'll come back and we'll show you where those parameters are in a couple of minutes when we get to that section. But this idea here of getting to the signal and reducing the noise by doing these heuristic approaches, this little windowing trick, and then that second bar, what that means is it slides the window along that many characters at a time, both save in computation because we don't have to consider every single character anymore, so we speed up our calculation, but they also help reduce the noise. Okay. And then our, what the off-diagonal runs of dots represent in this, these sequences here are the same sequence. So really we're looking for the diagonal run. The off-diagonal here are noise. That's what they represent in this particular case. if there was repetitive sequence elements in here, you would see those as runs parallel to the diagonal but off the diagonal here. And that's not the case. So really they represent noise in this case. And then the last question is really, why does this approach suck? Why is it not very good? And there are a number of reasons why it's not what we do normally to compare sequences. One of the main ones is, this is a visual approach, right? So I have to plot these things. I have to make this little figure and physically look at it. I could come up with ways of counting it, but I still have to kind of make it and plot it. And then it's kind of like, well, do you like it? Does it look nice to you, kind of thing, right? It's not a quantitative approach. It's not a number approach. If I had another two sequences different than the ones I'm plotting here, and I give you a different dot plot, you'd have to look at them and say, well, which two sequences are more closely related here? And it's kind of a, well, I like that one better, or I like this one better. And that's not a good, we want, I'm going to make you a little bit more nerdy with this class. I want us to put numbers on these things so we can unambiguously decide, no, these two sequences are more closely related than these two sequences because of this. Not a touchy-feely visual plot like this. We want to actually quantify these things and do it properly. Okay, so let me pause here for some questions. And Alina or Hangxin, if you can type in the chat window any questions that might have come up on Piazza. I don't have it open here actually, but I'll try and bring that up, and then we'll continue into section 2 of the workbook, of the lab sheet. Let me go here. Actually, I'll do that. There are no questions yet. Okay, excellent. I think that the dot plot approach should be pretty straightforward at this stage. So let's move on to the next section here, which is the Needleman and Wunsch alignment. So here, what we're going to do is I've given you two sequences and I've given you a scoring scheme, and we're going to use the approach that was discussed in the lectures, in the videos, to align these two sequences and find an alignment, find an optimal alignment score from filling in this table, as well as tracing back from that score, that happy place in the table, to find the alignment. So how do we do this? Well, let me return for a second to the here. So here we are. We've done our dot plot approach in our app, and now we're into this Needleman and Wunsch alignment approach. So the idea here is that we initiate these little tables, as I'm shown here for different two sequences, with our gap score. So we're going to go across that first row and we'll use whatever that gap penalty is, or gap score, and it's -2 in this case, as it is in the one in our lab sheet. And we're going to say -2, -4, plus -2 is -6, plus -2 is -8, and we fill in those initial rows and columns. And then the fun starts. Then we have to actually go and fill in that first major empty cell here in that top corner, the upper left here. And that's the only one we can fill in at this stage, because it's the only one that has 3 values all around it. Because what we need to do is, iteratively, we need to solve these 3 possible routes to fill in that value, the ones that are labeled 1, 2, and 3 here in the slide and the little blow up, and see which value is the highest. So we're going to consider all of these 3 values, the diagonal value, they're in root 1, so to speak, or straight down root 2, or straight across root 3, and consider what score is the maximal. We're looking at this in terms of the maximal score, and we're going to store that score in the cell, and also the direction that give us a little pointer to where we come from, or a little arrow that we'll use later when we go to find the actual alignment. So let's see, and in this case I filled it in, then we go and we fill in, we consider each one until eventually we filled in the length of our 2 sequences. We've got our optimal alignment score right there in the happy place, and then we trace back the arrows from that last cell, the one in the bright orange here, to all the way back to the start to get our alignment, as in this case. So let's go and see if we can do this here. Okay, so I'm going to try and do this on pen and paper first. I'm going to do this here. Let me turn this on, and we'll find my pen somewhere. Okay, so we have our 2 sequences. I've sketched them out here, and I'll maybe start, well, I'll leave it on black for a minute. So we've got 2 sequences. So what we need here is an n+1 by an n+1 table. That's our Needleman and Wunsch table, and I've drawn this out for you in the PDF, in the workbook, but I'm going to do it manually here, because this is what you would do, if someone didn't draw out the table for us. So I'm going to take that first sequence, and I'm going to put it down the first column here. So there's going to be a gap character to start with, because in Needleman and Wunsch we initiate it with these gap characters. So then I'm going to take my sequences A, T, T, G, let's zoom up a little bit, and then C. And then we're going to use that as our first kind of point of our cells. Then we have our second sequence, so there's going to be a gap character, and then it's going to be A, that's meant to be an A, G, T, T, C, so those are our columns. And you can obviously draw this neater than I can on this thing I'm still getting used to. So we have our grid. And this is looking a bit too small to put numbers in, but we'll figure it out. So our match score, let's write that in a different color here. Our match score was +2. Oops, excuse me, +2. And our mismatch score was -1. Okay, so that is going to be, if we ever go diagonally, that's that root 1, root 1, that's meant to be an arrow. Root 1 here is considering the match and the mismatch score. And then we have our gap score. So let's put that in a different color. So we'll do gap penalty, some people call it, but it's a score just same as the others, is -2. Okay, so these are the things we're going to consider. Now I'm going to go through here, and I'm going to take maybe red or green. Let's be patriotic and do green. And we're going to have to take that gap score, and we're going to start filling in our values. Okay, so in our case, I've actually drawn this slightly wrong. Let me see. Yeah, okay. So this is going to be 0, and then we'll do -2, -4, 6, 8, 10, and the same down here, -2, -4, minus, where am I getting this from? Well, I'm taking that gap score, that -2, and I'm adding it each time I go. Okay, so I'm taking this thing, -6, -8, -10. So I've initialized my problem, and I should follow my own advice, and I should store the little arrows. So let's put the arrows in. Those arrows should go down, 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 you know, across, 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 because I'm moving this way to fill it in. And anytime I move either right across, horizontally across from left to right, or if I move vertically downward from up to down, I'm using the gap score. I'm always going to use this gap score if I'm moving either straight across or straight down. I'm using that gap score. So now the fun starts. I get to fill in this value, and I have to consider my 3 possible routes. So here, you know, I can kind of look and see that it's going to be the diagonal route already, because if I consider going straight down, it's going to be -2 plus the gap score -2. Let's write this out to make sure that we're following along. And I'll pick orange here too, or purple. So if I was going to do the downward trajectory, downward route, I would do the -2, so that would be this value here, this -2, I would do -2 plus the gap score, which is -2, which would equal -4. And it would be exactly the same if I did it across the way. That would be from this value here. If I did this one straight across, it would be -2 plus again, and I go to my gap score up here, and I say -2. So this would be -4. If I'm going to go diagonally, you know, the route 1, it's going to be 0. So that's this value here, 0 plus, and now what I have to do is I have to look at the characters. And this is important because I'm going to use either the match score or the mismatch score when I move diagonally. I'm going to use either one of these two when I do route 1. And I have to decide which one to use. And in this case, this is an A, and this is an A. So I'm using my match score. So this is going to be +2. So this is going to be +2. And that is the value that I store. I've got my winner. And I take that value and I put it in, and I'll even try and stay consistent. I'm going to write +2, and I store my arrow. Okay? And now what I can do is I can move on, and I can consider either this cell, okay, or this cell. And the reason I can do only these 2 cells is because they have the 3 surrounding values. They have the 3 routes that I can use, 1, 2, and 3 to fill these in. So let's try another couple, and then we'll pause and we'll ask for questions to fill this in. So I'm going to go and I'll maybe do focus on this one, okay, and I'm going to erase this for a second. So I'm going to consider this new box here. So if I went and tried the going straight down, sorry, I'm getting terrible at drawing on this tablet. Okay, so it's going to be -4 plus, and I always use the gap score if I'm moving straight down, plus -2. Okay, so what's that going to be? Well, it's going to be -6 in this case. Now if I went straight across, I'm going to take this value, the +2 value, and I have to again add the gap score. So in this case, it's going to be 0 would be the answer. +2 plus -2 is 0. And then if I go diagonally, that's going to be taking this value up here. I'm going to have to take the -2 plus -2 which equals -4. So in this case, we have a winner. Our winner is straight across. So I'm going to go and try and erase this little thing out, and I'm going to fill this in with the green pen, and I'm going to put 0 in this box, and I'm going to store my droppings, so to speak. I'm going to store my arrow that came this way. Okay. So now what I can do is I can go through, and I can now either fill in this cell or this cell. And my objective, of course, is to fill all these in until I get to my happy place down in the bottom corner. This is, this is the place where our final alignment score will reside. This is the answer in terms of the similarity of these two sequences. Now, so if I did this for these two sequences and filled them all in, and I'll not torture you with my bad drawing and bad use of the tablet here too much, but if I fill this in for these two sequences and I got a value, let's say I'm just making this up, I don't know what the value is yet because I haven't solved it, let's say it's +4 is our final alignment score. If we compared another two sequences and it gave a value of +6, it would mean that those second two sequences are more similar, more closely related to each other than these two sequences. And I have an answer that I didn't have in a straightforward way with my dot plot approach. My dot plot, I just looked at it and I said, oh, maybe I like that one better. Maybe you like that one. This dots pattern looks prettier or whatever. Screw that. I've got an answer. +6 beats +4. Those sequences are more similar, more closely related, according to this, the input that I take, the scores for matches, mismatches, and gaps. So let's do maybe a couple more, and then we'll stop here and we'll leave it for you guys to fill this in and complete this. So in the PDF, of course, you can type in your numbers, but I do suggest you take a piece of paper or a tablet or whatever way that you physically write these things down. I'm only using this tablet because I have to show it online. Of course, I would use paper. It would be much easier and it would be much cleaner, and I can always erase it until you can figure out how to do these things. Okay, getting practice doing this sort of thing is going to be important. You should be able to solve this by hand, be able to fill in these little tables by hand. That's one of the major objectives for this week. So let's take another cell. I'm going to take this one here and try and fill this one in. So I'll erase my workings out, but I see the way I do it for every position. I don't know the answer beforehand, so I try all three routes. Okay, so I'm going to try down route first. So that would be taking the value +2. So by that I mean I'm taking this +2 value here, +2, and I'm going to take, in this case I have to take the gap score again, because anytime I go straight down, okay, so this is going to be 0. If I take the route across, that would be taking this -4 value, -4 plus -2, that's going to be -6. Okay, and if I went diagonally, okay, so that would be taking this -2 value, so -2 plus -2 equals 0. So I've got something kind of interesting here. I've got two possible winners, right? I've got this 0 and I've got this 0. They're both giving me the same optimal score for this sub-problem. And I'm finding these optimal sub-solutions to find the global solution to ultimately end up down here with the answer from building up these smaller solutions. That's the idea of this dynamic programming approach. So what do I do here? Well, I actually just keep both of them. The answer is 0, and my arrows are both. I'm going to store an arrow that comes from here, and I'm going to store an arrow that comes from, is that +2? Yeah, an arrow that comes from here. I used the wrong value, excuse me, I'm sorry. Let me go back. See, I made a mistake. I drew the square around it, but I took the wrong value. So let me go back. Take that arrow off. And this one, I did the wrong thing. I took the wrong value. I've wrote a big square around it up here. Let me just remind myself by drawing another square around it. I should have took -4. I apologize. -4 plus -2. Oh, no. This is -2. Yeah. I'm doing the wrong thing. I'm confusing myself. -2. It's this value that I'm taking. -2. So -2 plus -2. -4. Okay. So in this case, I'm taking that value 0, and I'm storing it. So let's do one more to make sure we're not totally confused here. Let's do this cell right here. And take that one. So you can see that one issue with this approach as we're doing it is that errors, like if I make an error like I did, it's going to be additive. It's going to propagate throughout the table. Okay. So what would you do in this case? Well, you know, the error propagates, you filled it in. If you've got and show the mechanics clearly how you got this answer, you'll still get most of the points, even if you don't get the right optimal score here. What we're looking at is kind of do you know the process, and can you think through this thing logically, how to get this answer? If you make a little mistake and it propagates through at this stage, we're not really going to penalize that in the homework or the labs. So let's do this square, and we'll try let's try the downward route. So we're going to take the -6 value here for this cell. So this is -6 plus, and we're going to always take the gap score, -2. So this is -8 in this case. Let me try and draw an 8 that looks like an 8. -8. If we were going to go straight across, this would be a 0 plus -2, which would be -2. And then if we're going to go diagonally, we're going to take this value, the -4, plus -2, -6, so we've got a winner, the highest value, -2, and I'm going to put that into the box, -2, and I'm going to store my arrow. Okay, so I've done the first little section of this, but you can see the idea. So if you fill in the rest of this, get to that final location down here, this happy place, we'll have our score for the alignment. That's what we're looking for. And we'll trace back our arrows through these cells. Okay. So let me pause. Oh, there's a question. Why do you add -2 for the diagonal when it's not a gap, rather match or a mismatch? Was that when I made the mistake? Sorry, I shouldn't. If I'm going the diagonal, so the question that's here, it's in the comments. It's from Nan, and she says, why do you add -2 for the diagonal when it's not a gap, rather a match or a mismatch? I shouldn't. If I'm doing the diagonal, my bad, I should use either the match or mismatch scores. If I'm doing the diagonal. The root one. Okay. So maybe we'll have another little review of this later if it's still confusing. It should be -3. Okay, sorry, I made a mistake. But the benefit here of having other people's eyes on it is that we would see this. So this would be this root here. Let me do it again. Right, this one. So I'm taking this cell and make sure we're doing this. So if we're going to do that, let's do the diagonal route, the one that I made a mistake of here. So we're going to take this diagonal route. So just to be clear, what we're taking here is the -2 plus, and now because we're moving diagonal, I'm going to take either the match or the mismatch score. And in this case, so I'm going to highlight, we're taking this character and this character, because this is the cell that we're doing. Okay, and the A and the T do not match, so I'm going to take my mismatch score. Okay, apologize, that was the mistake I made. I knew I made some mistake, but I'm trying to do too many things at one time here and confusing myself. Okay, so it's going to be -2 plus, in this case, -1. Okay, and that's going to, as the commenter rightly says, is -3. So that would be that root 1 answer in this case. Okay, so it didn't affect my, my final answer, because I was lucky enough that the downward root here is +2 plus, and in this case I always use the gap score, -2, which is 0, which still beats, this still beats the -3, so I still have my correct answer, and the cross root here is still -4 plus -2, which is no good either, right? This one is still, this one is still the correct answer. So I have the right answer, but I got it by making a sub mistake. Okay, hopefully that helps. So here, in this case, I'm still storing my arrow. Okay. Ali says, I'm a little confused as to how you got a value of -4 when calculating the first score, the box between the two A's. Oh, the only, where's the -4? So I'm going to zoom in a little bit for Ali's. So Ali mentions a comment, I'm reading it out here. It says, I'm a little confused as to how you got a value of -4 when calculating the first score, the box between the two A's. So the box between the two A's here in this case is, let's see if I can change color, is this box. This is +2. This box. So this is the A's and this is the A's. And so this gives the +2 box. Okay, let me take those off. So, and the reason I got +2 here is because let me draw that out. So if I was considering just that cell in this case, so I'm going to try and consider this. Whoops. I'm going to consider just that cell here. That's an A with an A in this case. So I'm going to try and draw that out one more time. So I'm going to consider the downward root, and that didn't work, right? And the solid root, but this root 1 root, the diagonal root, that was 0. That was the value that's here. Okay, so 0 plus, and the reason I took the +2 here to add on to it, okay, +2, is because this value comes from the match score, and the reason it's a match is because it's an A with an A, and those two characters are the same. It's an A with an A. So the tricky bit here, and the bit that I made the little oversight with too, is that you have, whenever you move diagonally, whenever you move in this kind of diagonal route, you have to consider either the match or the mismatch scores, and that depends upon whether the characters. If the characters are the same, you use the match score, and if the characters are different, you use the mismatch score. Hopefully that helps, Ali. And we can talk more in office hours too after the video. Okay, I'm going to pause here for any more questions, and I'll take this abomination of a piece of art off the screen and go back to our PDF, if that's okay. Okay, oh, Ali says, could you also do an example calculation for either the diagonal or to the right? Okay, sure, of course. So apologies for the delay. Let's go back and do that. Oh, office hours. No, let's do it, Ali. Let's do it while we've got it. We'll try and keep it open. So here's I'm back. So let me do the diagonal. We'll do one more diagonal, okay? And this will help. So, you know, whenever we try and teach this in class, if we're all together, we kind of use the whiteboards and we go around and we get people working together and folks correcting each other and explaining it in different ways and different words, and it really helps. You know, you've just got me at the minute using my words explaining these things. If you've got your peers there and listening and arguing and figuring it out together, it's the way you learn how to do these things. Like all of these little tricks and these math things. You know, this is like Sudoku on steroids a little bit, but the more you try it, and you try it with other people, or get feedback from it, from the IAs, for example, in our online session and with Piazza, the more you'll feel comfortable with it. So let's do, for Ali, let's do one more, and I'm going to here add one more horrible color to our screen. I'm going to try and do this cell here that I've highlighted in blue. And this is I'll look at the values here, and I notice if I'm going to try the diagonal route, which we'll maybe do first this time, is that it's a T with a G. So if I was going to use the diagonal, which would I use? Would I use the match or the mismatch score? Well, the answer here would be the mismatch score, because G and T are not the same, okay? So if I was going to take that route where I go diagonally, I would take the value in that cell that's diagonally here, so that's that +2 value. So it would be +2, and then I'm going to add onto it -1. +2 minus 1 equals +1. So that would be the answer for that route. If I was going to go straight down, I would take this value in this box, 0, plus, and I always, if I'm going straight down or horizontally directly across, I always use the gaps graph. That's the only thing I look at. So -2, so in this case it's -2. If I go straight across, it's another 0, so it's the same calculation. So I've got a winner. The winner is the diagonal route. So I'm going to go and stick that in the cell. So I'm going to write +1. That's meant to be a 1. And then I'm going to store my little droppings, which is my diagonal arrow. So if you don't store these arrows, you'll regret it later, because whenever we get to our final cell, our kind of happy place down the bottom corner where our optimal alignment solution is, we won't be able to trace back and follow our droppings through the woods, so to speak, to get back to the start, because this is the global alignment, it has to span both sequences from end to end. Okay, so I think I've given time there for folks to ask questions. I don't see any coming up on the chat window, so I'm going to move on straight away, if that's okay, and go back here, and we'll move. So there's 2 examples for you to do for that, and we'll move on to section 3, which is over to using BLAST. We have a protein sequence here from Fly, and we're imagining we're working with someone who's found this sequence in Fly and has been investigating its role in Drosophila or some other fly species, and they're saying, well, is there, this is interesting in flies, it does all these cool things in development and all these sorts of things. Is there something similar in humans, and what could that be doing? Okay, so we're going to take this sequence, and remember here I'm copying, this is a FASTA sequence, that's the format that's ubiquitous that we use in all these bioinformatics programs, and I'm going to take that and put it into a BLAST server. So let me go and pull that up. Okay, so here I am again, I'm back online, I can use this. So this is NCBI BLAST website, I'm just searching in BLAST, and in this case it's a protein sequence. So which version of BLAST do I use? I use not nucleotide BLAST, I use protein BLAST. Notice here in the middle we've got BLAST X, we haven't used it yet. But BLASTX, what it does is it translates nucleotide to protein. Remember we talked about our dot plot and how if you have the opportunity, it's a good idea to search for the protein. This does it for you. Right? This will have a lot more signal than nucleotide blast if you're doing that. So let's focus on the problem at hand and click standard blast P, protein blast. I'm going to paste in my fastest sequence. Here I'm going to make this window bigger just to make sure I've pasted this in correctly here. So I should see the sequence and no strange, funny characters or other things. And I've got the header line. I could give it a job title here. I'm just going to go and search non-redundant. I'll leave this alone. Maybe I'll exclude because he's looking for, can you find this outside of, or she, their collaborator. Let's say she's looking for things outside of fly. So I'm going to say fly here, and I'll say Drosophila genus. Yeah, that sounds similar. That's from the, that would search within the fly genus. I'm going to say exclude, and I'm going to run a blast search, blast P search. So these, anywhere where it had a non-default argument, it's kind of got my session settings from last time. I'm running blast P search, I'm excluding fly, and I'm going to search. So while that gets submitted and goes through, we'll wait for our results to pop up. And we'll have a little look back at our workbook to see what the questions are. So using the default settings for NCBA BLAST, can you find any homologs for this protein in humans? Well, actually we looked outside of fly. We used NR. So probably the answer is we're going to find lots more fly even outside Drosophila. Let's see. Well, I shouldn't jump the gun. I'm assuming that that's the case. Let's wait for our results. Okay. So if this is taking a long time, we can reload this page. Let's see if that brings it up. I actually have one here. So here what I did is I did a blast P search of the NR database, and it's amino acid 471. And what do I find? I find something called protein hedgehog, predicted protein hedgehog, hedgehog. And inside the square parenthesis, inside the brackets, here, if I zoom in a little bit, this thing, this name and this name and this name, they are the species in this case. So what are these species? Well, I'm going to pick one here. I'm going to copy it. And then I'm going to open let's open a new window. I'm going to paste this in, but just into a blast search. And I'm just the species name. If I had been able to copy that accurately, and I'll do a search. And I know that this is actually a fruit fly, Mediterranean fruit fly in this case. Let's pick a different one. Another species. Let's pick something further down, like something short, like this one, Sonic the Hedgehog, like the movie. I'm going to paste that in. I don't know what the species is. Oh. Okay. And this is going to be, it's not a Drosophila, but it's a species of skate in the family blah blah blah, found in Nova Scotia to North Carolina. Fish. So this is definitely outside of fly. It's in other fly. It's outside of Drosophila. It's in other flies, but it's also in these fish like species 2. Now let's see if we can edit this search and find it in humans. Okay, so here's the same search result back. I'm going to go edit search, and we'll focus in on their question. I'm going to go and change this to Refseq protein. Remember, reference sequence database is going to be less redundant than that one. I'm going to not exclude, but here I'm going to please search only within humans. So I'm going to type that in here, and there's two tax IDs. There's 9605 or 9606. So what the tax ID is, there's a taxonomy database at NCBI here, and we're using that to, all the sequences are annotated based on that in Refseq, and we're going to use it to just return results that are in human in this case. So I'm going to click on my search. I'm going to try and be patient for that. I also have the results over here. Let's see how this comes up. So here's one I prepared earlier. It's the same search. I'm searching Refseq protein. I'm limiting myself to humans. And I see there are 7 sequences in total in Refseq from humans. Desert hedgehog, Indian hedgehog, Sonic the Hedgehog protein, isoform 1, etc. I look at these and I see that the scores in the alignments range from 365 to 150. The query coverage for the first few is pretty good for 81%. I'll show you where I'm looking. I'm looking over here at the statistics for these searches. 81%, 78%, 71%, 64%. Again, what this means is that, in the case that I've highlighted, that 64% of that query protein, the one from our collaborator from Fly, that's matched in the alignment that we get here to this database sequence. So that's quite a large proportion. It's only 50% identity, but that's still pretty good. The key thing here is these e-value columns. We talked about this in the video lecture, and I'll encourage you to go back and examine that lecture again or rewatch it when we talk about e-value. This is really telling us here how likely this score, so a score of 277, would be to occur if the sequence in the database and the search sequences were jumbled and completely random with respect to each other. In other words, how likely is this to be complete gibberish, rubbish, a false positive, something that we're saying is related when it isn't related? Okay, so that's what the e-value is, giving us an assessment of the statistical significance of this score, of the alignment score, 277, like the score that we calculated in our little table that we were doing in the last section. This is the score from the local alignment, from the BLAST alignment. And this is not likely to be false positive. These are small e-values. We definitely trust these first 3 or more. And then we have pretty good e-values here too. So these are there are many sorts of things in this field, in the sequence searching field, are expressed as little rules of thumb when we consider these types of alignment statistics. People typically think of 25% or better sequence identity over a significant alignment span or coverage, where it covers the core, the heart of the protein you're searching with, the functional bit like the active side or the protein-protein interaction side, or if it's a nucleotide sequence, the transcription factor bind side, or something else, the functional bit that you care about in these sequences, that features in the alignment, that's in the coverage, and that the identity is up above around 25%. This kind of twilight zone that we enter in below that is kind of dangerous to infer commonalities of function. So what are these things? They have pretty good statistics. They meet that kind of criteria from the alignment statistics. I'm going to go and zoom out so I can see here. I'm going to click on one, like the first search result here, and the alignment, the texture of it looks pretty good. So it's 195 identical characters out of 389. So that's 50% identity, or positive, which means amino acids with a similar type, like where the pluses are here, like these sorts of things, like a serine and and tryptophan. These are conservative amino acid substitutions. We'll come back to what this means in a little bit, but they're not as drastic as being completely different character type, like a charged amino acid that's positive versus a negative or something like that. And NCBI results, I have this link out to the gene database that we visited last day and talked about in our videos as well. So this will tell me about the function of this protein in humans, what's known about it. So we can go back to our collaborator and say, yeah, we have found similar sequences in humans. Here's a listing of them, for example, and they do things that we're about to find out about here from visiting this gene database page. So if I actually click on that link at some point, it's opened in a new tab. So here it's opened in a new tab, sorry I didn't see it. So here it brings me to the gene page. I'm looking for the DHH, desert hedgehog, protein in humans. This will finally bring me to this gene entry for it. So here the official symbol is DHH. We write that down for our collaborator, desert hedgehog signaling molecule. It encodes a member of the hedgehog family. I can read this from the summary. Hedgehog gene family encodes signaling molecules that play an important role in regulating morphogenesis. Now I could read on and scroll down and look at literature links and things like that. What I suggested last time is that we use this thing called gene ontology here, this link out to gene ontology, because this gives this controlled vocabulary of functions and biological processes and locations where this thing is. And so here I'm looking at biological processes, cell fate specification, cell differentiation, cell-cell signaling, male sex determination. It's been linked to, as well as the things that it's involved in signaling, estrogen signaling, estradiol signaling. In these cases, it's found in the extracellular space, the plasma membrane. And I could explore each one of these kind of functions. As this is a protein, I would also go and I'm going to scroll all the way down to the bottom to find the link to UniProt. So here is the link over to UniProt at the EBI. So this is the premier protein sequence database. I'm going to open that in a new tab here. So here's UniProt, and it says DHH human, desert hedgehog protein. It's got the little gold star up here that it's a good entry, and the rating experimental evidence at the protein level. Intracellular signaling, essential for a variety of patterning events during development. And I could read more about these things. Again, it's the same Go processes listed here, like male sex determination. I could look and see, is there any pathology, any things linked to these things? I could do that on the NCBI gene page too. And here it's in developmental disease, like these ones, partial cause mutations, this gene affecting presence of testes on one side, or sex development, affected individuals of this karyotype presenting normally as females, but have no functional gonads. These sorts of things you could find out that might be of interest to your collaborator, for example, or dig into these things more. Okay, so that's the kind of way that we would explore this and answer the types of questions in this section of our workbook. So we can find lots of examples in flies, but also outside. Flies including in humans, like DHH. And we could write the names of the other proteins there. Here, select a few proteins and find the common name for this, for these. What trend do you notice as you move down the results? Well, you get the initial ones in that first search. By that question, what we were looking at here, when we look down this result, these first species are all fly-like things before we then, like here, this is like a mosquito, like let's see if we can copy its name here. Excuse me. Let's copy this and go to that web page. Let's paste this in, see what kind of species we get here. Yellow fever, mosquito. And you could explore that list, and you'll see that most of those top hits are mosquitoes and flies and other fly-like organisms. And before you then get out of mosquitoes and and out of flies, and then you get finally down to humans. And we really have to focus in to find those human homologs, or use those filtering options, because we're presented at the top with all of the list with all the things that are most similar to it. So you're going to find it in the closest organisms, in those flies. And then if you want to find it in humans, using that filtering that we were doing on our BLAST submission page. So when we did our edit and resubmit, we would find that we have to limit to the particular organism that we want to look in this case. So we're not drowned out, or kind of drowning in a sea of the most related sequences, which are all too close to the original sequence that we searched with. Okay, what were the other questions there? So here we did this, we limited to humans. What function do you think these proteins have? Well, they're involved in signaling. And we could show that they estrogen signaling, estradiol signaling, we could write all those things that we discovered here. Back in function, so we could take response to estrogen, cell-cell signaling. They're in the extracellular region. It's highlighted in yellow here, not intracellular. All these sorts of things, okay? So that's the idea of this. We're trying to use these questions to guide us. So some cell-cell signaling rather than intracellular signaling. And then, so list a couple of those there that you find out. What function do you think this protein performs in your collaborator's organism? Well, I would imagine that this, if we went back to our search results, we could find it, the one with 100% similarity. So I'm going back to the search results that we got when we looked at it. It's a protein, it's hedgehog protein. If we looked within Drosophila, here I excluded Drosophila, but if we edited that search and took this off, we would find that protein in Drosophila, and we would then link to the information about that protein in the gene database, for example, and we'd see that it's involved in signaling, cell-cell signaling, extracellular signaling, and cell-fate determination, morphogenesis, and these things. Okay, so that's the idea. We're using these search tools as a way to try and find out more about the biology. You know, what could be going on with these types of unknown proteins that we're presented with, or these genes that we come across in our research. So let's go back now, and we're going to, before we take a little break, let's have a look at this section, this optional section, section 4, the limits of using BLAST for remote homolog detection. So here we're returning to our good old friend beta globin that we used in our last lab, or we actually used in our first lab class, where we went and we saw a patient that had sickle cell disease and traced that all the way back down to finding that out and the molecular basis of that disease. So here we're going to take beta globin, and we know that in humans there are other globins, right? There's alpha globin that goes with beta globin to make hemoglobin, but we've also hopefully maybe heard of something like myoglobin and another globin. There's also neuroglobin in your brain, and cytoglobin, and a range of other globins that are present in humans. They're all homologs. That means they're descended from a common ancestor. And we'd like to be able to find them. We'd like to be able to align them and compare them and figure out their similarities. Okay. So let's go and use a BLAST-P search, Refseq protein, and see if we can find, for example, myoglobin or some of these other distantly related globins. Okay, so I'm going to go and go back to BLAST suite. I'm going to pick protein BLAST, and here I'm going to make sure that all these things are off. I don't have any of them. I'm going to do Refseq protein. I'm going to paste in my beta globin sequence. I'm going to make sure it looks correct. So I'll maybe give it a title, globin search in humans, and I'll limit myself in the organism field. Oh, I pushed return by accident. I'm actually going to go back because I didn't limit it to humans. So here I'm going to Homo sapiens, humans, BLAST-P. I might have to reload this because I accidentally pushed the return button. So let's go again. BLAST-P, organism, human, and search. Okay, so what we're doing, searching with beta globin against Refseq protein, but limiting ourselves to human results, and we're going to see what kind of results we get. What we'll find, and I know this because I obviously wrote the lab section, so we'll find beta globin, unsurprisingly, right? We find itself. These mosquitoes are bugging me here. It looks like there's mosquitoes coming to attack me. Let me turn that off. But we won't find the more distantly related globins. We won't find myoglobin, for example. So let me pull up a slide to show you what I'm talking about here. This was come back to this in a second. What I want to do is look at these globin sequences. So what are the globins in humans? Well, an evolutionary model of the globin family in humans, this over in the green box there, there's the beta globin, the one that we're starting with. And we'll find beta globins. Hopefully we'll also maybe find the alpha globins, that's in the light blue box here. And as we move further here, we're getting more distantly related, so less similar in sequence. And we might hopefully find myoglobin somehow, or cytoglobin, or neuroglobin. So neuroglobin is active in the brain and is thought to protect us from whenever we have oxygen deficiency and things like that in the brain. It's thought to be protective in that way. Cytoglobin, neuroglobins are more distantly related, more dissimilar than alpha globin and beta globin are to each other. So let's go back and look at our results. These have finally popped up here. And so here I can see it's a BLAST-P search. It's against the Refseq protein database. And we find just a few things, a handful of things, 9 sequences in my case here. I'm going to try and zoom in on those. I find hemoglobin subunit beta, that's reassuring, it finds itself. It also finds the delta, epsilon, gamma subunit. It also finds alpha globin, and it finds, the last one here is subunit mu. Note that there's no neuroglobin, no myoglobin certainly. Let's have a look at these statistics. Well, the first one, of course, is 100% identical, over 100% coverage, super low e-value, and these are all pretty good e-values. But you can see here that as we go down the table, the percent identity drops, the e-value gets larger, but the coverage is still good, e-value is still good, percent identity is above that, 25% kind of rule of thumb threshold. So we're all happy with these. We think these are all definitely globins. They're all related. They're all homologous sequences for sure. We can also infer that, of course, from the names, right? Hemoglobin, 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 they're all named the same. But I'm kind of disappointed because I didn't find myoglobin. I know myoglobin is a globin, so where is it? You know, what's going on here with this globin? Okay. So what can we do? Well, I'm going to go back and edit my search, and I'm going to use a more sensitive search approach here. So I'm taking everything's the same in this page when I click edit search, but I'm going to run SciBlast, this thing called position specific iterated blast. This will get into the next section of the worksheet, is why can regular blast not find these distant evolutionary relationships? And what can we do when we don't find them? Well, we use another tool like SciBlast. So let's run this and see what happens. And while I have that running, I'm going to go and describe what we're talking about here, or why this gives us an extra bit of advantage. I'm going to go back and pop to these slides and go back to those ones that I skipped over for a minute and say that. So we're when we were doing our alignment, when we were doing our little table and filling it in, that Needleman-Wunsch alignment that we were using, we were taking a match score and a mismatch score, one that I just gave you, a +2 and a -1, and these sorts of things. But I just invented those. They just came from the air, and we knew, as we talked about at length in the video lectures, that if we change the scores, we can change our answers. We'll change our values in the table, of course, but we can also change our alignment. Now, where do the scores actually come from? If we run BLAST, where does it get those match and mismatch and gap scores come from? Well, if we were on the BLAST page, and I'll show you this in a second, you can check on algorithm parameters and search or click on it and see where these scores come from. Let me show you this. So here's a BLAST page. I'll go back to the web site here. So I'm back on one of the web pages that we were using previously when we were searching for that fly protein. I'm going to go back and say edit search in this case, and it says here on right at the bottom down here, if you can see where my mouse is going right here, it's on algorithm parameters. So I'm going to click this box and it opens up, and it tells me that my word size is 6. That's my little window size that BLAST is using, and I'm using scoring parameters. I'm using Blossom62, and the gap score, there's going to be this. These are the gap costs. So it's telling me that it's going to score 11 minus 11 when it puts the gap in first, and then to extend it, a gap that already exists, it's going to be a little less. What is this Blossom62? What does this mean? Where does this thing come from? Okay, so that Blossom62, what that is, it's a table. So here it's Blossom62. It's a table that it uses, or a matrix that has scores in it for matches and mismatches. And BLAST uses this in its alignment scoring scheme. So if we're going to match, for example, a C with a C here, we would go up that first column, up to the top, and it would score +9. So all down the diagonal of this little table here, it tells me what the match scores are, because that would be the amino acid with itself, a character match. So the first thing to note is that not all scores are the same. So it's not like we were scoring +2 for every match. Some things like a cysteine score more than things like an alanine that only scores 4 here, for example. So if you have cysteines matching, that's more reward given than an alanine matching. That's because cysteines are rarer, and they're also usually doing important things in these biomolecules, for example, cysteine bridges and crosslinks and maintaining the structure of these biomolecules. So when we see these, and we like to see them conserved, we like to see them aligned, so we reward them highly. Now all the values that aren't on the diagonal, that aren't highlighted in blue here, if you can see them, they're the mismatch scores. We use this little table, we look up one row, we look across the other column, and we find the values. And some of those, the ones I'm highlighting in red here, they actually score positive. These mismatches are being rewarded. Others are scoring negatively. You can see a lot of negative values in here too, where the mismatches are definitely hurting us or penalizing us. So what are these positive scores? Well, if we consider one like this R with a K here, that means an arginine with a lysine. What this is doing, it's telling us that this R with a lysine, or R with K, this arginine with a lysine here in the middle, these amino acids, when they substitute for each other, they're likely to be doing similar sorts of things in the protein, in the biomolecule. They're conservative substitutions, and we want to award them. And that's because both a lysine and an arginine, for example, as shown here in the middle, they are both positively charged amino acids of similar-ish side chains, very similar character, and can often substitute for each other in the function and structural roles that they do in these biomolecules. Same is true of those aromatic amino acids above. This little table here is from a textbook, right? It's from one of those biochemistry textbooks, and it's showing the similar characteristics of those R groups, those amino acids that we would have. Another way to display this, although perhaps a little bit more complicated initially, is like a Venn diagram. Here the little groupings show us what's similar and what's dissimilar in these things. So that's all captured in these BLOSSOM 62 matrix. Okay, so BLOSSOM 62 matrix, it does better than what we were doing in that it doesn't just take one score and use it for every single amino acid regardless of what amino acids they are or what characters they are. It has an amino acid specific score, but it still takes that score and uses it regardless of where it is in the protein. So if it's at the start of the protein and not doing anything in the function, just hanging out and floating around and interacting with solvent, rather than in the heart of the protein, maybe in the middle, like here where my hands are, that does the function that binds the oxygen and does its stuff and transports it around, the amino acids that are there are likely to be much more conserved than the amino acids that are on the tail, but yet they get a similar score. And that's where a thing like SciBlast comes into its own. It'll treat amino acids differently depending upon where they are, and it'll learn that from our first search. So let's go back to our search results here, and this was my search results here. So this is SciBlast iteration 1 Refseq protein, and before I only had, I forget, was it 7 or so proteins that ended with this mu protein, but I've run this iteration of SciBlast and it finds a couple more. It finds cytoglobin, but still no myoglobin. So what I'm going to do is I'm going to click this run button again to run another iteration of this SciBlast program here. And this will go off and run another iteration. So it's going to take the search results that I've got in my first round and then search with those, with knowledge of those, to try and find more distant relationships here. And I'll explain what that means as this thing ticks over. So what SciBlast is doing, let's go back here for 1 second, is the purpose of SciBlast is to look deeper into the databases for matches to your query protein by employing a scoring matrix that's customized to your query. It's not this BLOSSOM 62 matrix, and it's not just a value of +2, -1, -2. It's a scoring scheme that's going to be a lot more sensitive to allow us to find more distant relationships, this profile, this PSSM. And it does this by finding the results of a regular BLAST search and then building rules to identify amino acids from those. Okay, so it builds this thing called a PSSM that what I should maybe do here is record a separate video about PSSMs for folks that are interested. It's kind of removed from our course. We're not going to examine it or test you on it in this iteration of the class where we're all in our learning online mode. But we would traditionally learn about this in the face-to-face class. It is an important method that we use a lot for finding more distant relationships. And what it does is it'll make a scoring scheme that's specific to your query from the results of initial BLAST search. So in this example here, we could see that it isn't just a +4 for an alanine-alanine match like would be in BLOSSOM, but that these matches will depend, the score will depend where it is in your input protein. So from position 1 in the protein all the way to the end of the protein, that's shown down as the different rows in this table. And then we have these scores that are either higher or lower depending upon where they are. For example, an alanine at position 6, that first red box in the first column scores a 5, but at position 15, kind of halfway down, it'll only score 2. And that's going to give us information again about where the heart of these molecules are. So if I run this SciBLAST, the way it works is we run a regular BLAST search, we construct a multiple sequence alignment from the results, then we build one of these models, and then we search, and then we iterate. We go around this cycle of iterating again when we click the run button. So when I do this, it's a bit like the idea here. So I'm starting with my beta globin sequence, this little red dot here, and I run a first search, and I find things like beta globin and alpha globin and epsilon subunit and things like that. And then I can use knowledge of all these things that I find to go searching again, to broaden my reach here. So I'm going to search now with this specific scoring scheme, this what's called a PSSM, that's the profile bit of the SciBLAST, PSSI, and then I iterate. And I'll find things, maybe my next search, like myoglobin. Let's see if we find that. And then I can build a new alignment and a new PSSM and search again and do an even bigger search. Now the problem here with this is that once I start finding things that are unrelated, something like this blue highlighted RNA polymerase here, which isn't a globin, but I include it into my model and go searching, I'm going to get corruption. I'm going to find things that aren't globins. I'm going to find polymerase-like things. And this is a problem. So we need manual oversight of this procedure. Let's see what it actually looks like here. So I did my first iteration. I found these things that I could find with regular BLAST. Then I run a second iteration. And let's go and see if we can find these things. We find new things. They're highlighted in yellow here, like myoglobin and neuroglobin. And then I can search again and find more stuff, and including things that are spurious, like in this case. Let me go back to our website and see what our second result, our second run has got. So here's my SciBLAST run, second iteration. It tells me that 37 sequences selected, sequences newly added this iteration are in yellow. So here we have it. Myoglobin and neuroglobin are now found for the first time in this approach. And they've got the little yellow ticks on them here to show that they're new. And I can go and I could look at their alignment and see these are 25% sequence identity, so they're right at that threshold. I could run again here and now see if I can reach even further, like that graphic we had a moment ago. So we're using knowledge of our hits that we find with an initial BLAST search to build an alignment, build a more specific scoring scheme for matches and mismatches, and then using that in our next search to go and see if we can find more distant evolutionary relationships. That's the idea with this procedure. So these topics are more advanced than the ones that we covered in our video lectures. They're optional, and for folks that are particularly interested in these kind of areas for their research purposes. I'll wait to see what results are returned here, and then we'll take a break. I think we'll come back and we'll finish off the last sections. Okay, so if the web server is being slow, like it is at the moment, we we can either go over to the EBI and use one of their BLAST servers, or we can just be patient here, the NCBI. But we're getting to the point where, you know, I'm getting frustrated with this web server being so slow, not returning results in time. So what I suggest is that, you know, subsequent bits of the course, we actually take control of these things back. We don't rely on other people's computers, that is other web servers here, but we take these methods back to our own machines and start learning how to do these things locally on our own computers. So in our next segment of the course next week, we're going to start our journey in that direction. We're going to start actually learning how to code things up and analyze our data locally on our own computers, and then go out to cloud computing resources when we need them. But we don't really want to be living in a world where we have to wait for a web server to give us back our results if we're facing some deadline or some critical question that needs to be answered quickly, as is often the case, right? So let's have a look at the last sections. What new hits were reported about their alignment statistics? So in this time we found myoglobin, neuroglobin, or distantly related globins, and we could add, here you would add details of the search statistics. That's the e-value, the percent identity, and coverage, for example. And I'll leave that as an exercise for you to do. So this is SciBlast. And then we have one, given the time, I'm going to say I'm going to leave this as an exercise for you to do. This is hammer search. This might also be a little slow given how the web's performing here. What we would do is, this is another method that's going to be more sensitive than regular BLAST. This uses a probabilistic model of sequences underneath called a hidden Markov model. We're going to go and take our sequence from our workbook and see if we can find those distantly related proteins. So here it is. Here's our beta globin sequence. I'm going to click submit, and if the gods of the intertubes are happy with this, we'll hopefully get some results back in a reasonable amount of time. It's thinking about it. So this web server can be slow. It's a lot more time consuming to use this server than others. This is at the EBI here. What we would find when this thing eventually works, if it will work, is that we get search results like this. Okay, so it's telling me, still parts of it are still loading here. I'm going to scroll down. So I've searched for this in it's found lots of hits. I didn't focus myself on humans, so let me go back, and it's found it's a globin. It's got lots of similarities. Hemoglobin subunit beta in different organisms. I'm going to go and try and replicate what we did in the SciBlast search. So I'm going to paste my beta globin again. I'm going to restrict myself here to let's do SwissProt database, and I'm going to restrict by taxonomy. I'm going to make sure I write humans in here, so it's restricted to humans. And then I'm going to go and submit. So here it's going to be more equivalent to the SciBlast search we were looking for. And here it's found similar to our BLAST results: beta globin, HBB, HBD, delta globin, epsilon. It also finds alpha globin, zeta, mu. It also finds cytoglobin, myoglobin, neuroglobin, all in one shot. Okay, and it says my search, when it eventually does load, was quick, 0.03 seconds. So this tool, this hidden Markov model tool, this is kind of at the forefront of the field right now for detecting these remote evolutionary relationships. These myoglobin, if is clearly a globin, we know that from our knowledge of biology, of course, that it is an homologous protein. We struggle to find it with conventional BLAST. We have to use these kind of specific scoring schemes that we talked about with SciBLAST here. And then in the last section of the workbook, what we do is, well, we look at the, so here we're all the way down to here, the divergence of protein sequence and structure. We use one last web server, one last tool here. I'm going to put this here. We use this tool called for structural alignment. So we're going to go and if this server works. Well, the web's not playing well today, so let's see if we can do. So the web server link that I pointed to in the workbook, this was working last week, but this is the nature of working with online tools. One of the big frustrations of them is, sometimes they go down, or sometimes they're not accessible, or sometimes our internet connection is too slow, as is my case here, because we're all working remotely, to get results back in an expedient way, or maybe even at all. And this person I know that wrote this tool, they were at Stanford Burnham, and they've left recently, gone to the University of California, Irvine. So I did a Google search to try and find an alternative server. I've never used this one before, but let's see if it'll work for us. No, this is just a link to the same server, which is down. Okay. So we're going to skip this section, I think, because the server doesn't work. We're kind of at the whim of the internet here. The idea here is we would search with beta globin chain. This is the 1-HBS or the 2-HBS chain that we used last day of beta globin. This is a myoglobin PDB structure, the 4-MPM, and we would do this. What we would see is these structures, which are superposed here. The top panel in the black background figure is the beta globin. The bottom one is the myoglobin. And when you fit these on top of each other, when you actually do an alignment on the structure level, they're pretty much identical. It's virtually impossible, or well, it's not impossible, but it's very, very hard to tell them apart. And that's telling us that structure is much more conserved than sequence. The structure retains those traces of common ancestry much more clearly than the sequence does. We really struggle to find those sequence similarities between myoglobin and beta globin, for example, with these sequence search tools. But if you have the structure, you can do this very expediently. You can pull up the structures if you're lucky enough to have them, or if servers like this work, and see these similarities very, very clearly. So it's showing this progression of sequence determines structure, determines function, and what biology really cares about is function, and so structure is more close to that, and we preserve the structure to preserve the function. So the sequence can often diverge and mutate and accumulate changes that render it beyond our kind of detection limits. With these conventional search tools like BLAST, for example, we have to turn to methods like SciBLAST, or even better, turn to methods like this hammer approach, this HMM approach. And we have to do a lot of work and be patient, and we can maybe find those things. Or we can turn to the structure, and if we understand a little bit more about structure, we can do these kind of comparisons and see these similarities in great detail. And we see that the conservation is there to maintain the heart of these molecules, as is shown in this graphic just here, where we've got the beta globin chain, and it's colored by its similarity or conservation across these hits that we got, like myoglobin and neuroglobin and cytoglobin. So blue means it's pretty divergent. It changes a lot going through into green and red, which is absolutely conserved. And the bits that are absolutely conserved are the bits that do the function. Those two residues that interact with the heme that carry the oxygen and coordinates the oxygen, those histidines, for example, those are absolutely conserved and present throughout. Okay, given the time and the slowness of the internet on my end here and these servers not being responsive, I think what I'm going to do is just pause here and stop here, and bearing in mind that these last 4 sections were there for extension purposes only, we're going to focus on those first 3 sections of the workbook, which was the dot plot parameter, approach understanding what the windowing does, what the stringency does, how that can help us get signal from noise. Then the second section was all about the alignment method. We focused on the global alignment method. You should hopefully have a better idea how to fill in those little tables and be able to submit that. And then the final section was using BLAST and filtering, how to get to results you might want. For example, we were looking for protein for a collaborator from flies, and we were kind of drowning in all the fly sequences, and we needed to filter that to really focus in on human homologs, right? We had to use those filtering schemes within the BLAST submission page. So those are the 3 main sections that we wanted to focus on today. We've gone above and beyond that in the last 40 minutes or more here. We've kind of hit our limit for attention span, I think, at this stage. So let's stop here. I'll pause and stay online for questions. We also have office hours at 12:30 via Zoom, and we have the Piazza discussion board where we can talk about these things in more detail. Okay, so with that, I think if no questions come through here, I'm going to finish off and say go all the way back to the, unfortunately there's no way to navigate these slides properly. I'm going to finish here. Okay, by leaving this globins that we were searching for in this case. Okay, let's finish there. Thank you. Hope to see you in office hours.